Hello, and welcome to this Integrated DNA Technologies webinar, Unlocking the Potential of Ultra-Low Frequency Variant Identifications for Cancer Research. My name is Ansha, and I'll be serving as moderator for today's presentations. The presentation will be given by our field application scientist, Jin Yao. He is a member of the IDT NGS group, providing support to researchers using IDT's range of NGS solutions in the field. He completed his bachelor degree in biological sciences at Nanyang Technological University and received a PhD in genetics at National University of Singapore. Prior to his current role, Jin Yao served as IDT's regional NGS applications specialist for the Asia-Pacific region and has research experience in the fields of the developmental genomics and immune cell biotherapeutics. Our presentations should last about 35 minutes. Upon completion, I will lead a question and answer session where we will address as many questions from the attendees as possible. Well, as a reminder, as attendees, you have been muted, and we encourage you to ask questions or make any comments during or after the presentations by entering your questions in the Q&A box at the bottom of the screen. Also, please note that you can expand the presentation slide window for easier viewing. Well, in case you need to leave early or want to revisit this webinar, we are recording the presentations and we'll make a link available on our website a few days after the presentations. You will receive a link in a follow-up email as well.
Thank you, Anne, for the introduction and everyone who have dialed in for today's webinar. My name is Jin Yao and I'm an NGS field application scientist at IDT. The topic for today's webinar is unlocking the potential of ultra-low frequency variant identification for cancer research. IDT has been supporting life sciences research for more than 30 years, and today IDT is a global team of over 1,500 associates, serving over 130,000 researchers in more than 100 countries. To meet the global demand for custom nucleic acids and genomic solutions, IDT operates production facilities in Singapore to support the Asia-Pacific market, in Belgium for the European market, and also 5 production facilities in the US. This includes our latest facilities in Ann Arbor, Michigan, and Boulder, Colorado, following IDT's recent acquisitions of Swift Biosciences and Archer, respectively. The XGen NGS portfolio offers a comprehensive yet flexible range of solutions which help streamline the sequencing process, whether it's for whole genome or transcriptome or metallome sequencing. In today's webinar, we will look at a unique chemistry that is designed specifically for sequencing of degraded and low-input cancer research samples. We will also see how this library prep chemistry can be combined with the XGen hybrid capture workflow to target cancer-related genes and identify cancer-associated mutations. If you would like to find out more about the various NGS solutions, please feel free to visit the IDT web pages or reach out to your local IDT sales team. Cancer refers to a group of diseases that can affect any part of our body. It is the most common human genetic disease where DNA changes or mutations in genes cause some of the body's cells to grow abnormally and uncontrollably. Cancer may develop when a single cell acquires a genomic change or mutation, which allows that particular cell to divide and multiply more rapidly than the surrounding normal cells, and this is known as the cancer stem cell hypothesis. This growth advantage allows cancer cells to undergo clonal expansion, during which the newly divided cells may acquire additional mutations that promote more invasive behavior or metastasis. Such mutations can result from errors during DNA replication or from DNA damage from environmental exposures such as tobacco smoke or the sun's ultraviolet rays. In certain cases, mutations in cancer genes are inherited, which may increase a person's risks of developing cancer. Cancer DNA sequencing is a powerful way for us to understand the genetic basis of cancer in order to develop more effective ways for cancer diagnosis and treatment. Next-generation sequencing has become the primary method for cancer DNA sequencing because of its high throughput in accessing multiple genes, even up to the whole genome in a single assay. NGS has enabled several omics technologies such as genomics, transcriptomics, and epigenomics, which allows us to analyze cancer genomes in a comprehensive manner, such as identifying point mutations, indels, and structural variations, including gene fusions. Now, this information not only further our understanding of cancer development and progression, but also enable the identification of actionable mutations or biomarkers, and using this information to guide the selection of targeted therapies according to the individual mutation profiles or to predict individual responses to particular treatment methods. Tumor FFPE samples and liquid biopsies are two common and important sample types used in cancer research. FFPE tissue is in fact one of the most widely used material for cancer research. It offers excellent long-term preservation of tissue morphology, leading to its widespread adoption and historical use for diagnostic purposes. However, the fixation and embedding processes often lead to DNA damage or DNA fragmentation and even cross-linking of DNA molecules to one another. It also greatly reduces the amount of tissue DNA that can be extracted, especially for rare tumor types. And even then, the DNA quality can vary greatly between samples due to variable fixation times or storage conditions. Now, liquid biopsy is another commonly used method which involves sampling of body fluids such as blood, and is a minimally invasive way of studying cancer by looking at cells or DNA fragments that are released into the bloodstream by tumors. However, because normal tissues also release cells and DNA into the bloodstream, DNA from tumors or circulating tumor DNA is present at much lower levels compared to a high background of normal genetic material. This low signal-to-noise ratio makes it challenging to detect circulating tumor biomarkers. And once cell-free DNA is released, it is exposed to various nucleases and enzymatic activities in blood, which introduces DNA damage and lesions. All these challenges makes FFPE and cell-free DNA inherently very challenging to sequence by NGS. In order to reliably detect cancer somatic mutations, an NGS workflow needs to provide high conversion efficiency. And what this means is having a high fraction of the initial DNA molecules being converted to sequencing-ready molecules. This increases the overall complexity of the library. We get lots of unique DNA molecules, including two here containing a cancer mutation. The high complexity translates to a higher sequencing coverage as we get more reads mapping to the target regions and therefore higher confidence in identifying low-frequency mutations. On the other hand, an NGS workflow with low conversion efficiency may convert, say, only 10 to 20% of the sample DNA molecules into libraries. This leads to libraries with low complexities containing few unique molecules, which reduces target coverage, even if we sequence to the same depth as the earlier example on the left. In this case, the assay sensitivity is reduced, mutations may be missed, resulting in false negative variant calls. So what are the factors affecting conversion efficiency and how can we maximize this process? Now let's look at the XGen cell-free DNA and FFPE prep workflow and the chemistry behind it to understand how conversion efficiency can be enhanced. The fragmented input DNA first undergoes end repair of any damaged DNA ends. We then attach a modified adapter through a highly efficient single-stranded ligation step. One end of this adapter contains a blocking group represented here by the red circle. This blocking group is critical because it prevents individual adapter molecules from being ligated to one another, thereby massively reducing adapter dimer formation. Now, the enzyme used in this step is an engineered ligase, which also eliminates adapter dimer formation. On top of that, it is also unable to ligate DNA inserts together, so the formation of these chimeric insert sequences is prevented. The combination of these two strategies favors productive adapter to DNA ligation, which maximizes the number of DNA molecules that get converted to libraries. Now, each adapter is also encoded with unique molecular identifiers, or UMI sequences. So these are added to both the top and bottom DNA strands during adapter ligation. The UMIs act as a barcode for the original DNA molecule, so any errors that may be subsequently introduced to the DNA can be identified and bioinformatically corrected. In this way, we greatly increase the confidence of calling out rare mutations in the tumor DNA sample. In the next step, a second adapter, shown here in blue, is added to create a double-stranded molecule. In the final PCR step, we incorporate sample index sequences and complete the library for sequencing. The entire workflow takes around 3.5 hours to complete, and the high library conversion efficiency makes it suitable for many demanding applications, especially cancer research that uses degraded samples for low-frequency somatic variant identification. So we wanted to find out how this workflow improves actual sequencing results. And to test this, we used 10 nanograms of cell-free DNA reference material as input with the XGen prep together with two other preps from supplier A and supplier B. On the left, each library was sequenced and the conversion rates and unique molecules generated from each workflow were measured. These are two key metrics for assessing library quality, and we see that conversion rates for the XGen cfDNA kit is around 3 times higher than A and B, meaning it is converting 3 times more input DNA molecules to libraries. With higher conversion rates, we would expect to get more complex libraries, and indeed, we saw a consistent trend when calculating the number of unique molecules in each library. And we then sequenced each library and look at target coverage, and this is represented here by the vertical axis against the read counts on the horizontal axis. What this tells us is that at the same sequencing output, for example, at 2 million reads, and looking at mean target coverage, we were able to obtain more than double the target coverage using this new adapter ligation chemistry compared to other traditional workflows. And the conclusion here is that a high conversion workflow can significantly affect the library complexity and final target coverage, and it allows the required coverage to be achieved with much less reads, which can help reduce downstream sequencing costs. One other important consideration when working with low-frequency variants is the presence of background errors that may be introduced at various steps of the workflow, such as polymerase errors or sequencing errors. These are represented here by the red circles, together with a true mutation in green. Now, these background error rates do become highly significant as we go down in variant allele frequencies of down to 1% or even lower, and they can mask the true mutations even with high sequencing coverage. This background error problem can be addressed by tracking each individual DNA molecule with a uniquely identifiable sequence, or UMI, which are color-coded here. And this makes it possible to link the sequencing reads back to the original double-stranded DNA molecule by grouping reads based on their UMI sequences. This allows us to identify errors which are sporadic in nature and then bioinformatically correct them so they are not counted as real mutations. Now, there is another problem though, which is that the UMI sequences themselves are also vulnerable to the same background errors as the inserts. And to address this, we have designed a set of defined UMI sequences and incorporated them in the adapter. In this way, even if there are sequencing or PCR errors within the UMIs, it is possible to identify and correct these errors to prevent artificial inflation of library complexities. This library prep and error correction workflow was utilized by a research group in McGill Genome Center who was working on biomarker identification in renal cell carcinoma using circulating tumor DNA from a mouse xenograft model. As renal cell carcinoma is a low circulating tumor DNA cancer, a high efficiency library prep workflow is required in order to sequence and detect tumor mutations with high sensitivities. The group tested the efficiencies of 3 library prep assays by generating NGS libraries from synthetic cell-free DNA controls, and they reported the highest library prep use in all replicate samples, with the XGen workflow shown here on the right in blue. After sequencing, the reads were processed once without the implementation of UMIs, and the per base error rate was calculated for different mutation types in blue. The analysis was then repeated, now with UMIs, to generate error-free consensus sequences for variant calling, and the error rates are shown in yellow. So we can see that using UMIs for generation of consensus sequences resulted in a substantial reduction in error rates for all substitution classes, as well as in indels, which really helped reduce false positive variant calls as demonstrated by this study. In this next section, let's look at how the XGen cell-free DNA and FFPE library prep can be combined with the XGen targeted sequencing workflow to focus sequencing and analysis only on specific regions of interest. During targeted sequencing, we isolate specific regions of the genome and then selectively sequence them so only a subset of the whole genome sequence is obtained. These regions may include protein coding regions of all genes through whole exome sequencing, or a smaller set of genes by using focused capture panels. Even with whole exome sequencing, we are sequencing less than 2% of the whole genome. So depending on what we are looking at, targeted sequencing can be very cost effective. It also produces smaller amounts of data, which makes data analysis more manageable and also reduces data storage costs. Experimentally, given the same amount of sequencing reads, targeted sequencing provides higher target coverage, which increases the confidence of calling out rare variants compared to whole genome sequencing. The two main target enrichment methods are hybridization capture and amplicon sequencing. Hybridization capture uses DNA probes to bind and capture regions of interest, while amplicon sequencing uses PCR to amplify DNA for target enrichment. In this webinar, we'll be focusing on the hybrid capture workflow, which generally works better for rare mutation identification. The capture workflow begins with genomic DNA or cDNA, which gets converted to libraries using any one of the XGen NGS library prep kits. The sequencing adapters are represented here in orange. Because the adapters are universal sequencers, they can lead to complementary base pairing and daisy chaining between library molecules, and this can greatly reduce capture selectivity. To prevent this, XGen blocking oligos are added to the libraries. These are designed to bind universally to barcoded libraries and can greatly enhance capture selectivity. We then add DNA probes or baits, which are designed to bind to the target DNA regions of interest. Each probe contains a biotin group, and when we incubate them with streptavidin beads, we can pull out the probe together with the bound DNA regions of interest. The remaining unbound fragments are then washed away, and what remains bound to the beads are the regions of interest, which eventually gets onto the sequencer. All XGen height probes are individually synthesized to guarantee high sequence fidelity. They're not synthesized on arrays, nor do they undergo any PCR amplification, and this helps provide the required on-target coverage and uniformity during capture sequencing. With a 4-hour hybridization, the entire workflow can be completed in a single day with the XGen height and wash kit. IDT offers a number of pre-designed and stock height panels, including the exome panel, AML, and pan-cancer height panel for cancer mutation identification. These panels can be flexibly supplemented with spike-in panels, such as the CNV panel for copy number variation analysis, or the human ID height panel to help track, for example, cancer sample identity during data analysis. Each and every single probe in these panels were synthesized individually and quality controlled to ensure consistent and uniform target capture and coverage. Besides pre-designed panels, we can help design custom height panels against the target regions of interest and then synthesize these panels with different options. The Excel panel is our fastest option, which can be delivered in as few as 5 business days. This is a good option if we need a panel quickly or for initial project stages where the panel contents are not finalized. The standard and production panels are shipped with an NGS functional test report to ensure a functional panel when you receive it. These panels undergo a pre-evaluation testing where they go through height capture enrichment and sequencing at IDT. The capture metrics for each panel are summarized in a report to ensure a working panel on receipt. This offering would typically have a few weeks of turnaround time. The production panels also ship with additional quality documentation, including individual probe ESI mass spec analysis. We also have recently launched the production panels without functional testing. Overall, the flexibility and options available means XGen height panels can be customized to any project to fit your research needs. Next, I would like to share a case study where the XGen cell-free DNA and FFPE prep is combined with the hybridization capture workflow to identify low-frequency variants associated with lung cancer. And this research study involved the analysis of lung cancer FFPE samples and matched adjacent fresh frozen normal tissues from 3 lung cancer subjects. Now, the sequencing libraries were generated using DNA from each set of tissues, and the libraries were captured with a custom cancer panel and sequenced. And by comparing variants which were present in the FFPE tumors but not in matched fresh frozen normal samples, we were able to identify tumor-associated SNPs, indels, and mutations. Together with germline variants, the workflow identified approximately 250 variants per subject. And these results were used to design XGen custom height panels that are specific to each subject. The panels range from 27 to 34 kilobases, depending on the number of variants identified. Finally, the panels are then used to capture cell-free DNA libraries to help us assess whether the subject-specific tumor variants can be identified in each cell-free DNA sample with high confidence. To do this, we generated libraries from plasma cell-free DNA from each subject and captured each library using subject-matched custom height panels. Following read alignment, the on-target rates were measured to be around 80% despite the relatively small panel sizes here. So majority of the data are actual usable reads that map on or near the mutations of interest. The high library conversion of the cell-free DNA and FFPE kits also produced high library complexities from just 25 nanograms of cell-free DNA. And this is represented here by the number of unique molecules in each library. The reads were then mapped and collapsed into combined read families using the UMIs on each strand to correct for errors in the reads. Because of the high library complexities, we were able to obtain high coverage of over 1000x consensus coverage for variant calling. After read mapping and error correction, we next performed variant calling. We were able to call out a set of subject-specific biomarkers at very low frequencies of less than 1% allele fraction, represented here on the y-axis. These are variants identified using cell-free DNA samples, and we compared them with variants from tumor FFPE samples from the same subject to determine sensitivity and positive predictive value, or PPV, of the assay. Here, the PPV is observed to be close to 100%, meaning we did not identify any false positive mutations, even at the low variant frequencies here. Next, let's look at how this workflow can be utilized for MRD research using NGS. MRD, or minimal residual disease, refers to the small number of cancer cells that may remain in the body after cancer treatment, such as cytotoxic chemotherapy. These cells can potentially grow and lead to cancer recurrence and disease progression. And often samples are collected from a liquid biopsy to study for MRD in a peripheral blood sample from cell-free DNA. This approach can allow a more precise assessment of treatment effectiveness compared to standard methods such as imaging, and help improve the accuracy of predicting cancer relapse. However, analyzing these samples can be very difficult as they are often degraded and at low input amounts containing only a few thousand copies of each gene. MRD can be detected using several technologies, with PCR being one of the most well established. This includes real-time PCR and digital PCR, which are often used for MRD detection as they are highly sensitive. However, PCR can only detect the specific genetic mutations that it has been designed to target, and it can be pretty time consuming if multiple mutations needs to be investigated. Flow cytometry is another common technique used to analyze cells in a fluid sample. It can be used to detect residual cancer cells by looking for specific markers on the cell surface. However, it is less sensitive than PCR, especially if the cancer cells have undergone significant changes in their cell surface markers, or if the cells are present in low numbers. And it can also be difficult to detect subpopulations of cancer cells that have a different surface marker expression compared to the main population of cancer cells. Now, NGS has emerged as a powerful method for MRD detection due to its high sensitivities and ability to detect rare genetic alterations. We can design targeted MRD capture panels for specific genes or genomic regions known to be associated with specific cancers. And this selectivity allows a cost-effective and efficient profiling of MRD samples and enhances the sensitivity for detecting relevant mutations and alterations. IDT offers a full solution for MRD research, which begins with a tumor-informed workflow. This is an initial analysis where we profile the tumor samples and match normal samples to identify tumor-specific mutations. Depending on the sample type and DNA quality, we can choose between different library prep chemistries. And this is followed by hybridization capture, typically with large panels containing a broad number of targets to find somatic mutations or unique signatures for each cancer type. Due to the larger size of the panel and higher sequencing depth required, this workflow is not used to identify ultra-low variants in MRD research samples, but rather it is used during the early research stage and discovery phase to identify variants that are representative of each specific cancer. And once we identify the specific mutations in the sample, it is possible to then look for these mutations at a later time and deeply sequence them for MRD detection. This is done by designing tumor-specific MRD panels targeting the sample-specific mutations and using them in a custom MRD research workflow. To detect the small fraction of ctDNA within the cell-free DNA sample, we use the XGen cell-free DNA and FFPE kit to ensure high conversion of the cell-free DNA to libraries. This is followed by enrichment for MRD-specific mutations using the customized MRD panels. In this way, the workflow focuses the library to a small target space for deep sequencing, which is required for ultra-low frequency variant identification. This is combined with bioinformatic error correction using the built-in UMIs to differentiate tumor-specific mutations from any background errors. The XGen MRD hybridization panels support flexible designs containing up to 2,000 probes per panel and are coupled with a simple ordering process that allows up to 50 panels to be ordered at once. And to speed up the workflow, all XGen MRD panels are synthesized and shipped within 5 business days from when the order is placed. And here's a comparison looking at how the XGen MRD panels can help overcome MRD research challenges more effectively compared to panels from another supplier. The XGen panels captures up to 2,000 targets, which provides 4 times more trackable MRD targets and ensures no target mutations are being missed out. And to simplify and speed up the ordering process, we have created a dedicated ordering tool that allows up to 50 panels to be ordered with just a few mouse clicks, and after which the panels have a proven turnaround time of just 5 business days from order placement to shipment. The icing on the cake is a 20% cost savings for a custom curated workflow that provides better value. In summary, the XGen NGS solutions are designed to help overcome a range of challenges in cancer research, whether for whole genome sequencing or targeted sequencing of cancer samples. During library preparation, we have seen how novel adapter modifications and enzyme chemistries can really help drive high NGS library conversion rates, which increases the overall library complexities and coverage that we can get from challenging cancer research samples. This is combined with UMI incorporation and stringent error correction, which increases confidence in variant calling at less than 1% variant allele frequencies. The library prep workflow is designed to work seamlessly with XGen hybrid capture probes and reagents to focus the library to a smaller target space for deeper sequencing through higher coverage while reducing per sample sequencing costs. And last but not least, the integration of these DNA technologies form the basis for the XGen MRD workflow. And researchers can now make use of this full solution for initial variant discovery from individual tumors, followed by custom target enrichment of these variants from low abundance cell-free DNA to identify the presence of circulating tumor DNA, which is indicative of MRD. And that concludes my presentation for today's webinar. Thank you for your attention, and I will now hand it over to Anne, who will chair the Q&A session.
All right, thank you, Jing-Yao, for that informative presentations. If you have questions or have not done so, please type it into the Q&A box. All right, there are a few questions in the chat box for you, Jing-Yao. So here is the first one. So I wonder, can I use my current adapters with the cfDNA kit?
Thanks, Anne. And no, we actually cannot do that because, as we have seen, the adapters in the cell-free DNA kits, they are not added through the traditional TA ligation step. Instead, we add them using a 2-step sequential ligation, which we have seen helps to reduce adapter dimer formation, so no adapter dilution is needed. But because the adapters are extensively modified specifically for this workflow, it will not work with other conventional NGS adapters. The XGen UDI primers that are used for the indexing PCR, they are universal with other XGen library prep kits. So those can be used interchangeably between the cfDNA kit and other XGen library prep kits. And for that, we can actually support up to over 1,000 indexes for sample multiplexing.
Thank you, Jin-Yao. I think there is a second question here. So I work in a cancer research lab. Is there any limit of detection supported by this workflow, particular for the variant identifications?
Yes, and in fact, we have been able to successfully detect variants down to 0.25% allele frequency with around 25 nanograms of reference cell-free DNA material. So one important consideration here is that rare variant detection depends critically on the amount of input DNA that we have. And if the input amount is too low, we can imagine the variant may not even be present in the actual sample. So when we are talking about low allele frequencies, the conversion rate actually becomes very critical to make sure that the variants actually gets converted to libraries and are being sequenced.
Thank you for the answer. And there's a third question here. So can you make a hybridization probe panel or MRD panel probes based on the design coming from another vendor?
Well, actually, yes, we can. And generally speaking, all we need for that are the actual probe sequences in order to make a custom panel. Or we actually have a free online tool and a very experienced NGS design team who can help us design the panels using inputs such as gene symbols or even date coordinates. And once the design is completed, we can actually perform a probe sequence analysis from your design to look at the on-target efficiency and also specificity. And what we do is we use IDT's proprietary probe design process to make a panel that provides broad and consistent coverage.
Thank you, Jin-Yao. Well, there is another question. So there is a functional testing on probe panel. What is it about and how is it being performed?
Yes, that is actually a great question. And the NGS functional test report is actually provided with the standard and production panels, really to ensure that the panel works as expected out of the box. And once the panels are synthesized, IDT actually performs a pre-evaluation of the panel performance through an in-house captures using the panel or using human reference material. And we will actually share the capture metrics for that panel in a PDF test report, which will be provided together with the panel itself.
Right, thank you for your answer. Yeah, I think, okay, that is all the time we have for questions. And I would like to thank all of you for attending today's presentations. I would also like to thank Jin-Yao for his informative sessions. As a reminder, we will send you a link to a recording of this webinar. Thank you again for attending, and we wish you the very best of your success in your research. Thank you.